Let's talk about protein modeling. We are mainly going to talk about homology-based modeling, meaning modeling proteins which are fairly similar to something which already is in PDB as an experimentally determined model. And so the things we are going to discuss now are how reliable modeling is at various levels of similarity, and we'll talk a lot about homology-based modeling, but then, the modeling competition CASP actually doesn't figure out in this talk now. But we'll talk about other methods in addition to homology-based modeling very briefly. And then I'll try to outline some reasons why we want to do modeling. Some of it is going to come in the beginning and more of it is coming in the end. So the most obvious reason for creating models is that it would be so nice to see a structure. A three-dimensional model, even if it's a theoretical model, helps you a lot in trying to visualize the protein and its functions, even if we know that it's going to be worse than a crystal structure. If nobody crystallized the protein and you don't have a crystallization lab yourself or a friend you could persuade for the project, so you just have to be satisfied with the models. And the other thing is that it's pretty expensive and slow to do crystallization and X-ray crystallography or NMR or cryo-EM. So it only happens to some selected few proteins. And so as of this spring, there were 160,000 protein structures in PDB. What is the current number, by the way? We might as well check. Do they have it in PDB? Most of the time they have it on the front. Oh yeah, it's like 172,000 now, so it's growing fairly quickly. But still it's less than 200,000. And compared to the 177 million in UniProt, again, we might want to check the current number. It's 209 million now in the Tremble section, and adding Tremble and Swissprot, it's almost 210 million. So there's a difference of 1,000-fold, and it's just growing. And even worse, we have lots of duplicates of the same proteins being crystallized over and over. Once you know the crystallization conditions for something, so it's so much easier to do a mutant of the same protein or try to crystallize a complex with an inhibitor or whatever with similar conditions. And if it's an important protein which was worth studying in structural detail, so often it's worth looking at more details like mechanisms of inhibition, the fit of specific inhibitors which might be pharmacologically useful molecules, and so on. So there are many proteins that have dozens, even hundreds of different models that have been done for various purposes. Obviously, nobody wants to do an exact repeat, but if you have a new inhibitor or a new mutant, so it's a good idea to see if any structural hints might help you in understanding what's happening there. And all of that taken into account, so out of the 160,000 models in PDB of this spring, so there's about 60,000 of different proteins. If you look at the statistics of 90% or 95% identical protein families. So you'll see that there's an average of a few copies of every sequence available in PDB. And because sequencing is so cheap, so the gap just keeps growing. And so if you want to see models, three-dimensional models of what we have in UniProt, so we have to be happy with the theoretical models. And here's a graphic presentation of PDB growth, and you can see that it was kind of exponential for a long time, like more than 10 years, and it's hard to see it, but it's more like linear growth for the past 7 or 8 years. And it's not slowing down because this represents just the 3 months of 2020. So there's a certain number of facilities for illuminating your crystals with powerful beams of X-rays, and they are working more or less 24/7. They have robotic handling of the samples, so that guarantees that everything will be changed immediately after the previous sample has been done, and the people don't have to be there 24/7. And you can see from the number of new structures in orange that it seems to have been pretty stable for, well, let's say the last 5 years. And the reason is that the so-called structural genomics initiative has expired, and there's no more funding to create more of these high-power crystallographic facilities, but instead the existing ones are working at full speed and still producing a large amount of new structures every week, every month, every year. But how can we do theoretical models? Well, this starts from the idea of knowing that anytime there's detectable sequence homology, if we can say that the sequences have a common ancestor, or a part of the sequence is from a common ancestor, let's say a particular protein domain or a module within a larger protein. So the sequence similarity always implies structural similarity, a similar folding pattern for the polypeptide. And because of that, so anything that has a related protein where the sequence similarity can be detected, in theory, it also can be modeled. And this is homology-based modeling. You have a target protein with a sequence only known, no known structure, and if you can find a structurally known homologue in PDB, so you are good and you can start modeling. And then in the absence of sequence homology, there are some curious ways of detecting sequence patterns without actually looking at the sequence, but just by looking at patterns of changes of, let's say, hydrophobicity, hydrophilicity, or the position of charged residues in a sequence, and so on. So there are fuzzy methods that can aim at recognizing the fold type even if the sequence similarity is too low. And this is not very successful. We have one more slide about that. And if there are no matches in fold recognition, it might be because your sequence represents a novel fold or is just too different to the known examples. So you have to invent new folds, or you have to invent new variants of existing folds to actually make a correct model for this very different sequence. And if you have to invent new folds, we are talking about ab initio methods, or from the beginning where you are just trying to create the three-dimensional structures straight from the sequences, maybe helping with a little bit help of some sequence motifs that we can recognize to be probably beta strands, probably beta turns, probably alpha helices. But still, this is kind of hard, and it's one of the eternal problems in protein biochemistry, the protein folding problem, how to figure out the way of folding a polypeptide knowing the sequence only, and it still doesn't fully work. So protein folds, you could say them to be synonymous to tertiary structure of proteins, the way of combining secondary structural elements together to create a globular pattern in three-dimensional space. And these protein folds also define protein superfamilies. You may have heard of, for instance, the immunoglobulin superfamily, which is represented in the antibodies, but also in hundreds, maybe even thousands of different proteins that have other functions. They might be other proteins in immunology. Many neuronal proteins are part of the Ig superfamily, and so on. And currently, these fold databases give us a catalog of about 1,400 different folds. But curiously enough, the discovery rate has just dropped to zero, so that no new folds haven't been really added in the fold databases. And so many of the fold databases just dropped out of business. There was no point of maintaining them because there was nothing novel to be discovered. There is still one which we'll see, it's called CAS, but still these classifications are quite valuable in annotating proteins and talking about proteins. It's a good idea to have names for different folds. And here are graphics of new folds. And this is cut so that the bottom part of the chart is here on the left. And you can see that the very early years were like not bringing too many models. But in the 90s or late 80s, the growth really began, so that people realized that structures are important, and the so-called structural genomics initiative started in the 90s, and you can see that there is a turn from a very slow growth exponentially into faster growth here. But if you compare this to the discovery rate of new folds, so you can see that when the structural genomics idea started, when they actually tried to find novel folds, so they were quite successful first. So in mid-90s, there was a lot of folds. Going towards 2000, early 2000s, again quite a lot, but then it started going down, and not so many new folds are coming up anymore. And let's talk about this structural genomics idea for a little bit before going back to modeling. So because we have homology-based modeling as a powerful tool, it's mainly the main limiting factor is not having models, templates for everything to start building the models with. So the idea was that they tried to find sequences that are as far as possible from anything which is known in PDB, try to crystallize them and try to get new kinds of proteins. And that would mean that when you have a wider structural space covered by all known structures in PDB, it means that you can also cover a larger fraction of the sequence space, which is represented by UniProt. And there are still structural genomic centers, but the protein structure initiative, the actual project has ended, and the prioritizing of strange proteins without any known functions attached to them has been mainly dropped. So now structural biology is more function driven, so that if you find an interesting protein that does something interesting, has some interesting properties, let's say it is a very fast enzyme in a previously known class, or is a very thermostable protein which can tolerate temperatures of 90 degrees in hot springs, or whatever reason we have for seeing a biological interest, they have to be the driver, and then you can start crystallizing. And if it's a novel fold or a very different sequence from anything known before, so it's a bonus. But if we look back at the graph of novel folds, so starting from 2009, there was very little, even if the protein structure initiative was still there. And so in the second phase, when they had completed the first phase of building up the infrastructure, getting so many structural genomics centers or X-ray facilities for doing these X-ray crystallographic structural experiments. So in the second phase, they just wanted to focus on crystallization. This was connected with a lot of developments in robotics, so that instead of having a PhD student or a lab technician pipetting away to create the 96 well plates with all the different conditions for crystallization, they were able to let the robots do that, and they created software for creating the crystallography screens, the primary screens for getting a large amount of different conditions. And if you had something that looked like a success, then you can do a tighter screen around the conditions that gave the initial success. And out of the centers, there were 4 that had to put 70% of their effort on the targets that were chosen because of the sequence dissimilarity, to get as broad a coverage as possible on the sequence space as 3-dimensional structures. But even with that, despite this very high energy put into it 2005 and 2010, this should have been continuing and giving as many as in the early 2000s, but it seems the sequence space, or let's say the protein fold space, was saturated, so that we just couldn't find any novel folds. So in the protein structure initiative, there were more than 4,000 unique structures, which were a structure of something that didn't have anything better than 30% similarity in PDB before. And even then, only a few hundred totally novel ways of folding a protein domain were discovered. And so why were there less and less novel folds? You might guess that the amount of stable protein folds is finite, so that maybe all of them are there in the 1391, which we know in a protein fold classification database. Or maybe there are still some, but they are so scarce, maybe we don't even know the organisms that are carrying them. It could be some strange families of archibacteria that haven't been discovered yet. And it also could be that when they labeled all the targets for investigating in this protein structure initiative. So anything that crystallized easily was achieved in the first years, and then it started taking longer and longer to create the crystals, and less of the experiments were successful. And that's another reason of moving into something that has a previously known biological function instead of just doing useless crystallization where you don't even know why you are doing it. But it also could be that the way that the crystallization conditions are screened are not optimal. Maybe there are some other parameters that should be tested outside of the standard starting setup. And anyway, anything that was crystallized in the standard robotic approaches with the known conditions for starting the crystallization attempt. So if it crystallized very fine, but you could talk about picking the low-hanging fruit. So they were just created quickly, and it got harder. And nowadays, not much effort is put anymore on proteins just for the sake of novelty if they are not characterized in any way. So you have to have some biological data. And still another guess of why you don't get any novel folds is that when the fold space is being more densely populated, you are running into problems of deciding what is a novel fold and what is a variant of an existing fold. So the fold databases are kind of, even back in 90s, they had subtle differences in deciding on how to classify the folds. They were broadly kind of in agreement, but the details are differing. And knowing that the fold databases are also not so actively maintained, they are not getting the funding, so we might have a few or maybe a dozen novel folds waiting in PDB which just need to be classified. Or maybe it is a combination of all these, but we could say that we have run to the end of the saga of discovering more and more different protein folds, so that we know probably a great majority of all the different ways of how to fold a water-soluble protein. Back in 1980s and 90s, it was thought that because there was a similar saturation of new folds coming in, it was thought that maybe 1,000 is the magical number, that there are no more than 1,000 ways of folding a protein. But with the protein structure initiative, the number of folds turned into an increase again, and so people stopped guessing at the numbers. So I wouldn't say that 1,400 is the maximum. Maybe it's 1,500, but the truth is that we don't know. We only know that we probably have the majority of them. And if we are talking about proteins in vertebrates, or probably even in plants, so they have been investigated much more intensively for biological reasons, and everything in the human proteome can be modeled now based on homology-based modeling. So the whole sequence space of human protein folds is already covered. Well, maybe there are some tiny domains somewhere, but let's say the major domains in every protein are modelable. So with a long introduction in the previous slides of having different folds that are probably all discovered, or most of them discovered, it does make sense that there are fold databases for classifying and cataloging these folds. And actually, that was one of the things that was needed for the structural biology, structural genomics projects too. And just to make a small diversion of what is a protein domain. So domain means a structural unit, as a unit separated in space from the rest of the protein. And crudely, you could say they are like beads in a string. So kind of globular units linked by a flexible linker or hinge unit, and then each domain could be crystallized separately and studied in the structure. And also it is considered in terms of protein folding that folding takes place within each domain. So most of the contacts are within the domain and very few contacts are formed, or much less contacts are formed between domains. And many of protein motif databases have developed motifs which can detect single domains of specific types, and they are really a powerful tool of discovering the domain borders in existing proteins or figuring out the number of similar repeated domains. And these motif database representations are kind of prototypes or abstract representations of these domains, and they are really good tools. And the PFAM is one example of such a domain database. And these PFAM sequence patterns for different families are one way of defining protein folds. Interpro is another one, but Interpro includes PFAM and many other resources as well. And sometimes a protein domain can be assigned a specific function, so that, for instance, in many protein kinases, we have the kinase domain, which is separate, which carries out the catalytic function, and then we might have other domains which are helping in anchoring other proteins or deciding on the protein localization of the kinase, etc. And also, most of the time, a single domain is coded by a set of exons. In the extreme case, it could be a single exon that codes for a single domain. But anyway, we can have these exon collections easily shuffled, and that's coming up here. So having exons and introns is something that all eukaryotes have, and even, well, actually, I'm not sure if any prokaryotes do, but having introns is something that makes it very easy to make recombinations of DNA with two different domains put together. If you copy a piece of DNA that contains the exons coding for a single domain and move it to a new environment, it's very easy to add a new exon into, sorry, a new domain into an existing protein. And these genome rearrangements could then create new functional proteins or new multifunctional proteins by combining two different functions into a new exon combination. And to give you an example of these complex domain architectures, here's an Interpro architecture which has 1, 2, 3, 4, 5, 6, 7 different domains, and this particular combination of these domains is found in 38 different proteins. And here's a structural example of a protein with two domains. It might be hard to see it in the space filling model in the bottom, but when you look at the ribbon diagram, so here would be the borderline between the two domains. You can see that the beginning of the sequence forms this domain, and it doesn't no parts of the polypeptide are part of the second domain except after here. So it's kind of rare that we have a continuous beta strand here combining the two domains instead of having a linker, and this is a very rigid combination so that the domains are not like beads on the string, but they are rather like two beads that have been welded together. But both of these have a similar Ig superfamily fold. You can see beta sheets here, which are like two pieces of bread in the sandwich, and unsurprisingly, this is called the beta sandwich fold. And here, the other beta sheet at the back is here, and the other one, which is in the front, is a little bit harder to see. It's got only 3 strands and a small irregularity here, and maybe these 2 strands are part of that sheet as well. But anyway, this is still another beta sandwich. But this is a 2-domain protein, and you can see that this is like a repeated domain. We can detect the domain boundaries by sequence analysis and so on. And this is actually a history of things already gone. We could say that SCOP and FSSP are efforts that are already dead, no updates in 10 years, but CAT has had a last major update in 2017, but nevertheless, they are still releasing new analysis tools, and all the new models of PDB are being classified into existing CAT categories. And there is CAT-B, which has these automated daily updates, and it's not as well annotated as the main CAT, but still the website is alive, and even if the CAT itself is not under active development, it is run by a structural bioinformatics group, which is releasing other tools which are making use of the existing CAT resource. So it's a good idea to have a look at that. We have a link to that in Moodle, so feel free to look at that. And now, after a very long digression, we can jump back to modeling. And let's talk about what uses we have for the models, again, to keep our motivation fresh and be excited about modeling. And the models, I like to call even the PDB structures as experiment-based models. They are not the absolute truth. We've seen all kinds of mistakes and imperfect R factors which are associated with the structures, so they are only the best interpretation of the imperfect diffraction data that we have, or the imperfect NMR data. So they are the models which satisfy the largest possible fraction of the data items, or largest fraction of the structure factors, bringing us a minimal R factor, or the maximum amount of structural restraints in NMR. But anyway, the PDB models most of the time are known as the real structures, and they certainly do have a much closer connection to reality and a justification because they are backed by experiments. And then there are the theoretical models, which is the main topic today. And whether we have an experiment-based model or a theoretical model, when we create a visualization, so they are really a big help to the scientists and even to the general public. It is so much easier to explain how something works when you can show it in pictures. You can show that, look, this is where the oxygen is bound in hemoglobin. And so it stays there, and once you arrive at a lower pH, or a lower partial pressure of oxygen, so the oxygen gets released and so on. And for the scientists, seeing the target of their research in three-dimensional detail is a big help in figuring out what might be important. So you might want to create mutations that you engineer into the protein to see if your ideas of important residues are correct. If you have a disease where you have a mutation in a protein which is too scarce to purify from the patient, so instead you can create the mutation in the lab and produce the protein and study the activity and do the crystallization and see it. And even if you don't crystallize the mutant, trying to figure out if the mutant could be involved in the function of the protein is something that is helped a lot with the visualizations. And then when we have the three-dimensional shape of a protein, we can try to fit different small molecules to this model. Drug docking or chemical docking is really popular with pharmacologists. With pharmacologists, they try to see if in which way the drug might fit in the active center, or in the absence of known drugs, they might use the docking experiments in planning a drug. So that would be ligand docking when you are looking at how a small molecule is landing into a protein. And we can also try to model protein-protein interactions by trying protein-protein docking. But that's like a lot harder than ligand docking. But this is not the topic of this course. It would be really lovely to learn about methods of docking and their limitations, but we'll just stay at being happy to say that docking methods exist and are a good reason for creating models. But now this computer-aided drug design was not as big a success story as people wished. So it seems that in the time you spend in trying to create a couple of rationally designed drug candidates, you are not as certain to have them working as drugs. So the success rate was not good enough so that it was worth paying for months of somebody's salary to create these candidates. So instead, now people have gone back to the combinatorial chemistry. So once you know what kind of pharmacophores you want, you can define that, okay, we need to have a negative charge here, we need to have a hydrogen bond donor here, which is 5 to 6 angstroms away from the charged residue and so on. So giving these chemical identities and distance restraints between them, you can start creating molecular structures just with computer software, and giving the additional limitations of having a sufficient solubility in water so that you could have people taking these drugs either orally or as injection. In both cases, you need water solubility. So in that way, you can easily create 10,000 different candidates by combinatorial in silico chemistry, and then maybe synthesize 1,000 or 2,000 of them and do high throughput screening for those compounds to actually see which of them are inhibitors or binding to specific proteins. And that's a more productive way of getting drug candidates. Okay, short course in drug design over now. So let's go back to homology-based modeling, and this is an important slide in the content in that it gives an idea of how easy it is depending on the sequence similarity. So you need to have a template protein with a known structure and then the target protein which is similar to your template. So in practice, you take your target protein sequence and you run a blast search on the sequences of PDB proteins. And the identities you get, the higher the better. So if it's more than 70%, so we know that the model is probably going to be without any grave errors, and the process is fairly easy. We can rely on the model. Even 50% is good, so that the models are doable without major problems. But then at 30%, you are not even sure if your sequence alignments are going to be fully correct, and then of course the logic fails and you don't get a reliable model. And in the twilight zone, under 25% of similarity, sometimes you might get lucky even at 20% similarity and get something that is probably correct, but this needs some special help, some landmark residues you could know pretty convincingly before you can rely on the alignment. So this is a graphical presentation of what you try to do. So when you want to figure out the structure for a sequence you are interested in, you find a template, you align the sequence of the target and the template, and then you leave the identical residues as they are, and whenever there is a change, so you change the side chains, this methionine to isoleucine, etc. And this is an easy example in that there is just well, there are two gaps here. So I'm not sure how to approach this one. Maybe you could leave out the gaps and decide that you model the end in the same way as the other one. But whenever you have insertions or deletions, you have to think carefully how to do those parts. And the biggest problem in homology modeling is that the results tends to be more like a caricature of the real thing than a very precise photograph. So you can never have a model which is closer to the true structure than the template is, and most of the time it's hard to create a model which is very different from the template. So they tend to be just very similar to the template. And then if you really don't know which parts in your sequence you align with what parts of the template, you are really on thin ice. And you might try to help it. You might try to test your alignment accuracy by constructing multiple sequence alignments instead of just a pairwise alignment, which you had here. So let's say we have 30% identity between the target and the template. And if we can create a multiple sequence alignment which has some sequences which are 50% identical with both the template and the target, or we have some sequences 50% identical to the template, 50% identical to the target, and then we have some other sequences which are, let's say, 50% identical or 70% identical with these bridging sequences. So there is less room for alignment error there when we have more sequences as data. And if you are unsure of the correct alignment, even that, you can then create alternative alignments and use each of them for a separate modeling project and try to see which of the results is most realistic. Now, homology modeling is certainly the best method there is, the most accurate way, and some people say the only meaningful way to create models, but the guys at Google would disagree. There is a new guy in town called AlphaFold that can do template-free modeling at fairly great accuracy. But anyway, it's not available for the general public, so we have to satisfy with the servers we have. And two of the high-ranking softwares and servers in modeling competitions or modeling assessments are called iTasser and Modeler, and Modeler is something we can use in exercises in this course. There's another one called Swiss Model, which has a nice collection of ready-made models, but it hasn't been ranking very high in the recent competitions. And the makers of Modeler have made a huge collection of models called Modbase, and the good news is that we can access Modbase directly from Chimera. So we just have to know a UniProt accession number to a sequence of our interest, and then we can search very quickly if there are any models in Modbase corresponding to that. And now, what do we do if we don't see significant sequence similarity? Is it even possible at all? And sometimes it is, because there are rare cases where the structural similarity is still there, even if the reliably detectable sequence similarity is long gone. So some superfamilies have extreme members that only carry a 9% sequence identity so that you can't align them. But when you put the proteins side by side, any biochemistry student who's looked at protein models probably can tell that, oh yes, they look pretty much the same. There's an extra loop here and something missing there, but it's the same fold. And you can do a superimposition with software which could tell you the same thing. But then it's not really detectable by sequence alignment methods, because if you are going to 20% sequence identity, so you can create a lot of non-homologous alignment matches. So they are just disappearing into the noise. Even if you can align them, the alignments are not statistically significant anymore. So for this twilight zone of sequence similarity, very dim sequence similarity, you certainly need to have other methods. And fold recognition, also known as threading, when you are trying to thread the polypeptide sequence into a specific way of folding. So it's based most of the time on sequence profiles of physical properties instead of relying on amino acid identities. It's vaguely similar to sequence comparisons. You could think you are lumping you are lumping amino acid identities together into classes and doing kind of a low resolution alignment. But anyway, it's not using the sequence similarity directly, and it can benefit from a combination of let's say polarity properties and size properties, which is not directly in the sequence methods. But still, it hasn't been really successful. And if you want to look at it, there's a site called genesilico.pl, which is a consensus method taking its input from multiple servers with different approaches and trying to do its best. Well, sometimes you need to combine methods. So if you have a homology model that has long insertion, let's say 30 residues which do not exist in the template, and they are in the middle of the sequence. So you might try to do an ab initio or de novo prediction for that part of the sequence, or maybe there's a terminal extension of 10 or 20 residues. You might try that as well. But then again, the terminal sequences tend to be structureless anyway, so this might be futile. So whenever you have these problematic methods, it's a clear and simple way, and often justified, just to leave them out. So instead of trying to model your 30 amino acids, you can just draw a dotted line connecting the ends of the additional domain. Or you might indicate a gray transparent sphere representing the size of a globular protein of 30 residues, just to give an idea of the volume which is missing from there. Now, the models we build by homology methods probably could be improved. I mentioned that it's hard to go beyond the template so that they become a restriction to the model, and you would want the models to go a little bit closer to the real thing. And you could try to do model evaluation and try to see if there are some problematic areas that are not good in terms of rules of protein structure, and try to rebuild them, or do things like energy minimization or protein dynamic simulation to create some movement into the dubious parts and try to see if they snap into place into a more favorable conformation, and maybe improving. So you can do these modified models and try to score them and choose the best one. But the problem is that if you don't have the real structure, you can't use a structural similarity-based scoring function as they do in the modeling competitions. So how to design a scoring function to represent the goodness of fit to something you don't know, it's kind of hard, and it's like Baron Munchausen trying to pull himself off from the swamp by lifting himself from his pigtail. So the best we have is just try to look at the credibility and feasibility of the structures with these model validation algorithms and model validation scoring, like the Ramachandran plot. But this is still an area of active research in structural biology, and it has been a category in the critical assessment of structural prediction. So they give you the best model after or best few models after a first round of predictions for a specific target, and then your task is to try to improve the best models. But it hasn't been hugely successful, so the methods are certainly still lacking and they are still needed. And finally, the ab initio methods. This is the same Latin words that they use in chemical modeling, but the ab initio in chemistry means looking at the electron orbitals and all the quantum mechanical effects and getting a computation that gives you precise bond lengths and bond angles based on physical principles, quantum chemical principles, but you can't go that detailed with proteins that have thousands of atoms. So instead, the beginning they go to is just the rules of protein folding, principles of protein structure, trying to find sites which might work as nucleation sites for an alpha helix formation or nucleation of a beta strand, which are the two beta strands that are most likely to snap together first and work as a scaffold for further folding and so on. And then you can do brute force simulations trying to do energy minimization of the candidates and trying to improve them in that way. And sometimes novel folds have been discovered and proven to be correct later when the crystallization has happened, but it is still like very rare and a bit speculative. But anyway, if you're interested of something more, so remembering the name Rosetta is good. You may know that Rosetta Stone is a famous archaeological artifact that helped in deciphering the Egyptian hieroglyphs, because it was a stone that had translations of the same text in hieroglyphs in the cuneiform script, which were these arrowheads, and then some third script of ancient Middle East, and that helped as a key of figuring out the meanings of the hieroglyphs. And in the same way, the Rosetta folding project is trying to go to the very basic rules of protein folding and go to the abstractions of what defines a good protein, so that it could discover new proteins, new folds which were not known to exist. But then there is a new artificial intelligence approach, but it requires a lot of computing power. It's been only running in Google's DeepMind computer and artificial intelligence system, and they really cleared the table in the last CASP competition. And we have a link to the abstract describing AlphaFold and how it works, and it's certainly a promising method, and it would be nice to have it in the use of the general public. Okay, in addition to running Rosetta in your computer or the guys at the Rosetta site using the servers they have, there is a project of Rosetta at home where you can share your computer for computing some little parts in the folding problem of whatever targets they have, maybe participating in cancer research. And the idea is that it's a screensaver. When your computer is idle, you are letting the processor run the computation of them. It's the same approach as they had in the SETI at home, search of extraterrestrial intelligence at home, which is trying to find signals in the radio telescope signals they are scanning scanning from radio telescope to see if there's a pattern, if it's perhaps containing a message from somewhere. And this Rosetta at home has been good in that there have been many nearly correct models of novel folds, and some of that has been even in the CASP competition. And then finally, there's a way of using the power of crowds instead of the power of multiple professionals. There is a video game called Foldit, which is for manual optimization of folds or trying to do protein docking and so on, or fitting into the electron densities of crystallographic experiments. And they come in the form of competitive puzzles, and they have very quickly usable tools which are doing crude protein dynamics runs or helping you to minimize the structures. And if you have an eye for the protein structures as a biochemist, or if you have an eye on how to play this game without knowing anything of proteins or biochemistry, you can get good results, and they have a pretty good energy scoring function, which is what gives you your place in the high score list. And many of these high scoring models created by the Foldit high scoring players have been actually producing, again, near correct models that are for proteins never seen before and verified later by crystallization. So this is really fascinating, that you can let even non-experts have their hand in protein folding. And I never went into the competitive puzzles myself, but I went through the introductory targets of learning how to use it, and it was pretty addictive. I could imagine going for it at some point and doing this hours and hours. So it's certainly great fun, and when it's also science at the same time. So what better can it be? Okay, so finally a few words on what are we using the models for. So what specific parts in the visualization can help us. So one thing is looking at the environment of each residue in the sequence. So when we see the protein in the folded way, we can see that, oh, these residues 99 and 100 are not that far from residues 210 and 211. So we see that there are contacts between them, and if we are changing something out of them, maybe we are making the structure in that region of the protein a little bit weaker. Maybe it's decreasing the stability of the protein. We can make hypotheses of the consequences of changing something, and it's fairly easy to do this site-directed mutations to create the sequence and produce a recombinant protein, purify it, and test it. And even if you don't do the mutations, you might understand the functioning of your protein better when you see a picture which you can rotate yourself and see where the active site residues are, where is the substrate of the enzyme, and so on. And of course, if you're thinking of immunology and antibodies, so if you want to create an antigen related to a specific protein, so having a model helps in planning, so that you don't want to create a peptide-based vaccine which has only core residues which are never exposed in the protein. It wouldn't be helpful in recognizing the foreign organism. And then we did the exercise of coloring a protein based on sequence conservation, and that is really nice in understanding the function of the protein. You can look at each of the highly conserved residues and try to figure out why is it there, why does it have to be that particular glutamic acid. and comparing the observations in your own eyes to the articles that have been written on the significance of specific residues. So it gives you a much deeper understanding of a protein, and if it's something you are going to be studying yourself, so you are at a much better footing in making informed decisions and thinking correctly about the protein. And then finally, you might want to look at how your protein behaves. If you simulate a short period of its movement, you might be able to see what parts of the protein are not moving that much and what parts do move. And so in that way, for example, in my favorite protein, carbonic anhydrases, they have verified the hypothesis that a specific histidine number 64 can work as a proton shuttle, so that when the histidine is moving around and it's flipping from towards the center, towards the outside conformation, it can pick up a proton near the active site, and by flipping it can release it outside of the protein. And the simulations are able to show us that both of the flipped outside and flipped inside conformations are favorable and fairly highly populated in the simulations, so giving a higher reliability on this hypothesis, which was based on static structures only in the beginning. And now if we have a model, does it tell us really how the protein works? If we don't know the active site based on experiments, it's nearly impossible to identify it from a theoretical model. If we have a three-dimensional structure, an actual crystallography-based model, there are some features in the model, like having the PI value of, the pKa value of a specific ionizable group changing from the theoretical. So that's an indication that it's an active site residue. But these pKa changes do not happen in homology models. They are not precise enough to reproduce these effects. If we have several experimental structures or sequence similarity to narrow down our search of important residues, it's getting a little bit easier, but anyway, it's not that easy. And if you are thinking of something like the binding of steroids in a steroid hormone receptor, so and thinking of which residues are important there, so these simulations can give us some answers. We can see which residues are there and which residues bind tightly, which residues are nearby but are free to move. So we can do a lot when we have models, and I'm still uncertain if I should create a small talk on protein dynamics, which would tell you more about how these simulations are run, but we'll see. But anyway, if we have known features of the protein, so if we know what is the substrate binding site, if we have a PDB model with the enzyme and the substrate crystallized together, we can see what are the substrate binding residues. Maybe the mechanism of catalysis is known through experiments, and we know which residues are there. Looking at them in the 3D model is pretty cool. Likewise, if we have a ligand which is not really in the active site, we can look at the details of its binding, like looking at the binding of of protein of glucose 2,6-phosphate, which is a regulator of hemoglobin oxygen affinity, and it's binding outside of the oxygen binding site. And then if we look at conserved residues which are not in the actual substrate binding site or enzyme active site, we might be able to assign the odd ones as critically important for the structure. We might see that, okay, we have a glycine which is conserved and it is in a beta strand. Okay, it has to be the smallest amino acid to allow for the polypeptide to make that bend. And trying to study any of that and trying to explain it after you self make the hypothesis that, okay, we probably have these prolines needed for structure, so it's much easier for others to understand when you have good quality figures, as we can prepare by UCSF chimera.